Welcome back to Chapter 11, Part 3. In this, we're going to deal with a few ideas that Mendel had no idea about, but his principles were spot on, and nothing that I am going to talk to you about in this set of examples violates what Mendel talked about. It's just that, he didn't know everything, and that's okay. So the first thing that we're going to talk about is how Mendel didn't have everything exactly right when it came to dominance. So when we talk about Mendels concepts, we talked about dominance and recessiveness. Well, I'm going to use a slightly different version of that same saying. I'm going to say complete dominance. So complete dominance is what Mendel learned about. He knew that. Complete dominance occurs when the phenotype of the heterozygote and the phenotype of the homozygous dominant are the same exact phenotype. That's what complete dominance means. Heterozygous or homozygous dominant look exactly the same. But it turns out that that's not always the case. There's something called incomplete dominance. And with incomplete dominance, the F1 hybrids are kind of in the middle between the two parental variances. So for example, if you have a red parent and a white parent with incomplete dominance, something in the middle would be pink, right? Because pink is like halfway in between red and white. So we would use the term incomplete dominance. Then there's this idea of codominance. And codominance says that if you have two dominant alleles, the effect on the phenotype, they both show up in the phenotype, basically. They're both showing up. So with codominance, what you have is both a red and a white flower. So here we have the striped flower right here. So with incomplete dominance, same red and white parent, but you get pink, something in the middle. Incomplete dominance. If you've got the red and white parent and you see stripes or polka dots or something like that where they're both showing, that's codominance. If you did this cross and they all showed up red, then you would have complete dominance. Hope that makes sense. But those are very important terms as we're going along, and we're going to talk a little bit more about that in detail. Let's just take a quick look at an example of how you would write out a problem that shows, in this case, incomplete dominance. So because we don't have complete dominance, notice that our alleles look different. So if we had complete dominance, we would use big R and little r. That's for complete dominance, but we can't use those because we know that big R stands for dominant and little r stands for recessive. When we have incomplete dominance, we can't do that. So instead, we use a two-letter lettering system where we use a letter, usually the letter C if we're referring to color, and we kind of do a superscript and we have it raised to another letter. So C raised to the R is saying that that allele codes for a red flower. Okay? So over here we see C raised to the W. The color is white. So this one would be a white flower. Now, it's so important that we use these alleles because if we used capital letters and lowercase letters, then we wouldn't be able to really just glance at it and say, oh, they're talking about incomplete dominance or codominance. So we use different letters for allele names. Again, it's this combination of C raised to the R or C raised to the W that makes the allele. So here is a homozygous red and here is a homozygous white. CR crosses with WCW. They can only make one kind of gamete each. The red one will make CR gametes and the white one will make CW gametes. When you do a CR combined with a CW, then you are going to get pink, in this case, of incomplete dominance. It's heterozygous, CR-CW. Now remember that we can do statistics, we can use probability. So this pink flower will make half CR gametes and half CW gametes. And if we crossed it with another pink flower, right, they would also make half CR and half CW gametes. So a quarter of our offspring would be red, 1/2 times 1/2, and a quarter of our offspring would be white, CW, CW. But 1/2 of them, 1/2 times 1/2 is a quarter, 1/2 times 1/2 is a quarter, and you add them together. So 2/4 of them would be pink, CR, CW, heterozygous. How does this work? It's such an interesting thing that you can get things showing up in the middle. And so there is a relationship between dominance and phenotype. And when I say dominance, I could be talking about complete dominance or incomplete dominance or codominance. And so let's take a look at this and look at it at different levels. Some of these levels we haven't talked about yet, but make some sense to us, hopefully. So let's dive into this a little bit. Remember that alleles are the variations or the various forms of the gene, right? But remember that the gene is DNA, and DNA is made up of nucleotides. So if you have two different alleles within one individual, right? You'd think that they were heterozygous because this is allele 1 and this is allele 2. Maybe this is allele big A and this is allele little a. Or that would be normal dominance. Or maybe it's incomplete dominance with CR and CW, right? We've got two different alleles is the whole point here. If they're showing complete dominance and it's existing in an individual that shows recessive, then they're heterozygous. And one important thing to remember is that these two alleles are not actually interacting with each other. This one goes through its own DNA makes RNA and RNA makes proteins and proteins have functions, and that's how it shows up in the phenotype of the organism. So that's how it shows up in the appearance of the organism. So this allele isn't even touching this allele. They have nothing to do with each other. This one is just happens to be the dominant because it shows, and this one is not dominant because it's not showing. Literally, that's all it means. Forget about the incomplete dominance for now. I'll get back to that in a second. Right? Okay. So if you have a gene and it's on a chromosome and you've got one allele, and let's be real specific here. Let's take an example from a disease, and this disease that we're going to talk about is Tay-Sachs disease. This is a fatal disease. It's caused by an enzyme that doesn't function. Okay, it doesn't work, and specifically, this enzyme is found in the lysosomes of your cells. Remember that lysosomes are digestive bodies within your cell, and they're filled with enzymes. And those enzymes will break down whatever substrate that they interact with. In the case of Tay-Sachs, the enzyme that is not working properly is the enzyme that breaks down lipids. So if your cells aren't able to break down lipids, then those lipids will accumulate in parts of your body. The worst thing for people with Tay-Sachs disease is when those lipids accumulate in the brain. That's just a terrible thing because your brain can't break those down, and then you end up dying because you have all this lipid basically shoved inside of your brain. It's just stuck there. So if you're heterozygous for Tay-Sachs, then you make both the normal allele for the enzyme and you have the Tay-Sachs allele. And what does that mean? Like, what does that mean that you have the Tay-Sachs allele? Well, you have this DNA and there's a mutation right here. This should look familiar to you because I was talking about this with purple and white earlier, but I've changed it to be something real specific that you can kind of wrap your mind around here. The normal allele for the enzyme is going to be made by the normal DNA, and it's going to produce the normal lysosomal enzyme And if you can make this enzyme, then you don't have Tay-Sachs. You just don't have it. But imagine that you had two alleles for Tay-Sachs. Just really quickly, the allele for Tay-Sachs has that mutation, and the enzyme it's just not even produced because this mutation is such a bad mutation that it prevents the enzyme from being produced. But that's okay if you're heterozygous because you're still making normal enzyme. But if you're homozygous for Tay-Sachs, which is a recessive disorder, then you can't make the enzyme. And that means that you'll die as a young child. Most people with Tay-Sachs die by age of 2. So if you look again at the whole person, if they have Tay-Sachs disease, then they're recessive. And the allele for Tay-Sachs is recessive because it's not making the enzyme. Right? As long as you make the enzyme, you're showing the dominance. So that's at the whole organismal level, we say the allele is recessive. But if you look at it at the biochemical level, this is the level of the actual enzyme, we might say that this is incompletely dominant because the enzyme that's being made, it's only being made about half as much if you're heterozygous. This allele, this gene's doing fine, it's making that enzyme just fine. This one is not. And so this one can only make half as much as if they could both make it. And so what you have here is something that's incompletely dominant. You're only making half the amount of enzyme, but you know what? That's okay. Because enzymes don't get used up when they're made. They just keep doing their job. So at the biochemical level, we would say that you're incompletely dominant just because you only make half the amount of the enzyme. At the molecular level, we would say that those alleles are co-dominant. And they're co-dominant because they are both going through this process. They're both showing up, right? They both exist as genes. So they're co-dominant. It's just a different way of looking at it. We tend to look at things at the level of the phenotype. And so we tend to refer to Tay-Sachs as a recessive allele. Now, other important things to think about is that just because we're saying dominant does not mean that that means very abundant. There are lots of instances where dominant alleles are either deadly or very, very uncommon. Here's two examples that I'm going to step you through. The first one is that did you know that, here's a child with 6 fingers. 1, 2, 3, 4, 5, 6, right? Here's another one with 6 fingers. That's called polydactyly. And we think about 6 fingers as being rare. We think about 6 fingers as being uncommon. And we think about it as being recessive. But 6 fingers is actually dominant. If you have the allele for polydactyly, you will have 6 fingers. That's all there is to it. You might be homozygous or you may be heterozygous, but you will show 6 fingers. 1 in 400 babies in the United States are born with an extra fingers or toes. It's rare, but it's dominant. Another example of that, I'm sorry, I thought I had it on this slide, but I don't. Another example of this is in Huntington's disease. I think I have that a little bit later in my lecture. Huntington's disease is a dominant disease. And if you have the allele, then you will have Huntington's disease. If you don't have either of the dominant alleles, if you're homozygous recessive, then you will not get Huntington's disease. Let's put this all together in a very practical way. And the thing that we're going to talk about here is something called multiple alleles, but we're also going to be bringing back our idea of codominance. So let's just start out with talking about what multiple alleles means. For most genes in a population, there's actually more than 2 alleles. I know it's confusing because we've been talking about dominant and recessive, right? And just thinking about 2 alleles and how we make those gametes. But for sure, there's a lot of variation in organisms, and part of that's because there are more than two alleles that exist in the population. It's so important that you understand that I'm talking about the population level and not you as an individual. You still just have two alleles for each gene, but you and I might have very different alleles depending on what we're talking about. Again, blood type is a really good way to start, and we will talk about blood type here. There are 4 phenotypes of blood type in the ABO blood type group. Now, you might also have heard of positive and negative. We're going to ignore that, okay? Just for now. We'll talk about it a little bit more in lab, but for now, there's the ABO blood group, and there's 4 phenotypes. There's A, there's B, and these are phenotypes, right? There's AB, and there's O. But there are only 3 alleles, and the alleles are named after their enzyme, and their enzyme starts with an I. So we use the letter I and raise it up to a superscript. That should sound familiar to you. If you're raising something up to a superscript, it must mean incomplete dominance or codominance, right? Okay, in this case, it means codominance, and we'll get an idea of why that is in a second. All right, so I raised to the A will do something really specific. It'll put a carbohydrate named A onto your blood, onto the outside of your of a protein on your blood. If you have the B allele, the I raised to the B, I superscript B, you will put the B carbohydrate on that blood. If you have the lowercase I, now remember when things are lowercase, that means they're recessive, then you're not going to put any carbohydrate on the outside of that blood. So what that means is that if you're type AB, you must have both the A and the B allele type, IA, IB. If you're type A, if your blood type phenotype is A, you could be 2 different genotypes. You could be IA, or you could be IA little I, because you could have one of the alleles be recessive. Or you could be homozygous for A. Same thing for B. You could be homozygous for B, or you could be heterozygous for B. You can only be heterozygous for AB. It's not even possible to be homozygous, because you have to have one for the A and one for the B in order to show both A and B. And for O, you actually have to be homozygous. It can only be little I, because the second you get a big I, A, or a big I, B, you would be that other blood type. So you have to be homozygous recessive if you're blood type O. How does this work? Again, you're putting these carbohydrates on the outside of the blood, and so this is what the blood kind of looks like. If you're type A, your IA allele is going to put that little triangle-shaped little cartoon here for you, little triangle-shaped carbohydrate on the outside of that cell. If you're type B, you're going to put the little circle one there. It's just a carbohydrate that's being shown as a circle. And if you're AB, you're going to put both the triangle one and the circle one. And if you're type O, there's nothing there. Literally, O in this case means nothing. Zero carbohydrates on the surface of your blood cell. So we could do some genetics problems with this, and we will do that when we get into lab. But for now, let's take a look at a final concept here, and that is, well, I guess it's the second to last concept here for the strangeness that is Mendelian genetics, and that is Pliotropy. Now, I just said Mendelian genetics, and I said Pliotropy. Mendel didn't know anything about Pliotropy. Mendel also didn't know anything about multiple alleles, but that's okay. So when we think about Pliotropy, we think about how a gene can have multiple effects on the phenotype. It's only one gene, but that one gene, again, 2 alleles maybe, but one gene multiple effects on the phenotype, that's Pliotropy. So, for example, if you have a disease called sickle cell disease, or maybe you have a disease called cystic fibrosis, you have difficulties not in one thing in your body, but multiple things in your body. So let's take a look at some of the effects that come with having cystic fibrosis. With cystic fibrosis, you have salty sweat, and we're not just talking about like normal saltiness. It's very salty. You get frequent lung infections. Your heart is enlarged. You have terrible pain associated with gallstones, difficulty digesting food, your pancreas doesn't work well, and you have trouble breathing. That's 1, 2, 3, 4, 5, 6, 7 phenotype effects. But it's only a single gene. So the same thing happens, and this is what we're looking at for the idea for sickle cell disease. If you have sickle cell disease, again, you just have the one gene, but you have many different effects. Like if you have sickle cell disease, you might develop blindness. You might develop liver failure, and you are susceptible to heart attacks. So one gene, many effects, Pliotropy. One gene for cystic fibrosis, many effects, Pliotropy. So we've been talking about, earlier we were talking about like dihybrid crosses, and that's very interesting, especially if you're just looking at dominance. But when we're talking about 2 or more genes, there's some weird interactions that could happen. Remember before I was saying, let's just pretend that they don't interact. Well, they can interact. So we'll talk about how those genes can interact when we're take a look at some Labrador Retriever genes. Or alternatively, we could think about how many genes could affect a single trait, like as in human skin color. We got many, many, many genes that go into determining human skin color. So let's take a look at our Labrador Retrievers first and get a new term called epistasis. So epistasis happens when a gene at one location, one locus, alters the expression of a gene at another locus. So Labrador Retriever coat colors, you might know there are black labs and there are yellow labs and there are chocolate labs, right? It actually takes 2 genes to determine coat color. 2 genes. It looks like a normal dihybrid cross, but you only have one characteristic that you're looking at, and that's coat color. So one gene determines whether or not you get color. So in this case, brown or black. Big B for black, little b for brown. Just by looking at this, you should be able to know that black is dominant and brown is recessive because we're using big B's and little b's. But there's this other gene, and it's the gene that we're calling the C gene. And the C gene codes for whether or not the color is deposited or whether there is no color in the hair. If there's no color in the hair, you end up with yellow. So let's take a look at this, let's take a look at this. So if you're big B, big B, big E, oh, I'm sorry, those should be E's right there. Let me fix that for you really quickly. Okay, it's fixed now. So if you have the big B allele, you'll be black, and if you have the little b allele, you'll be brown. But if you have the big E allele, sorry for that fix, you will either be black or brown, whatever one you say, whatever one the other thing says, you'll be. E says, yep, big E says, yep, you'll have that. But if you have the little E allele, you don't get black or brown, you just get yellow. So let's take a look again at some of these. If you're big B, big B, big E, it means yes, you'll have color, and the color will be black. Yeah? Big B, little b, big E, yes, you'll have color, and that color will be black because black is dominant to brown. Let's look at a brown one. big E, little b. Yes, you have color, and that color is brown, recessive. Let's take a look at one of our yellow ones. Little e, big B. Okay, so no color. If you did have color, you'd be black, but you don't have any color because you got the two little e's, so you're yellow. Same thing here. You'd be black there. Here you'd be brown because recessive, but you don't have the big E to say yes to make the color. You have the little e's, so you're not making the color, so you're yellow. When you do crosses like this with epistasis and a dihybrid cross, you get this ratio of 9 to 3 to 4. That's very common. In human skin color, things are a little bit different. Instead of being, you can only be 3 colors, right? Like in the Labradors, it's black, brown, or yellow. That's it. That's all you get. With humans, you get like a whole range of colors. Look at all these beautiful colors here. Anytime you get a full range of things like this, be it color of skin or color of hair or height or weight, things like that are quantitative characters. They vary along a continuum in the human population or in any species population, but we're talking about humans right now. So if you have quantitative variation, that usually indicates, not always, but it usually indicates something called polygenic inheritance. And polygenic means many genes. So instead of having one gene or 2 genes, you have many genes, polygenic. And all of those genes together kind of add up to give you a specific effect on a single phenotype. Again, the human skin color is the example that we're stepping through. So if you have 2 like really white people together, that they will have like only white babies, right? So, what it happens though, if you have 2 people that are completely heterozygous at all of their 3 genes in this case, that means that each of these people can make a whole slew of different gametes. Each of them could make a whole slew of different gametes. Basically, you can have 8 different gametes if each parent is heterozygous. And so we can use those and we can kind of like do our multiplication. 1/8 times 1/8 is 1/64. So if both parents are heterozygous at each gene, they have a 1 in 64 chance, and you can see that down here, by the way, of having a very, very, very pale child. On the other hand, those same parents have a 1 in 64 chance of having a very dark child. And most of their children are going to be somewhere in the middle, but look at the possible combinations that you have for a child that's born with a skin color somewhere in the middle. That's why you get all these amazing shades of skin color in humans, because it's not one gene that codes for skin color, it's many genes, and they all affect the same trait of skin color. Polygenic, quantitative characteristics. So far, to this point, we've only been talking about genes, right? I mean, of course we have. We're talking about Mendelian genetics, right? But we can't leave this conversation without talking also about how the environment affects the way that individuals look. So, this is that argument of nature versus nurture, but we're really looking at the way that individuals look in their appearance. And it turns out that environment as well as genetics affect the way you look. I mean, if we're talking about skin color, right? Let's think about skin color and the environment effect on skin color. If you have medium color skin and you sit out in the sun, you can get pretty dark. But if you're like a really super pale, there's no way you're not going to get very dark. So even though they might have experienced the same environment, these two people, they're going to have different expressions of that. But that said, both people do respond to that environment of being out in the sun by having their skin color change as a result of that. So the range of phenotypes that can be, it can be very broad for polygenic characters. That range of characteristics, it's called multifactorial because it's not just genetics that influences it, it's also environment. So we'd say multifactorial. Genetics and environment affect it. Hey, you know what? Genetics and environment affect everything. Like everything. Everything is pretty much multifactorial. You just have to think about it. So just to wrap this part of the chapter up, an organism's phenotype is its appearance, outward appearance, sure. It's also the internal structures. It's also physiology, like enzymes and all of that. It's also the way an organism behaves. An organism's phenotype reflects its total genotype as well as the environmental history that organism exists in. All right, well, that's our wrap up for chapter 11, part 3. We're moving on to really just focusing on how we can figure out genetics in humans in a little bit. And that's part 4, human genetics. And we're going to be looking at pedigrees to help us figure that out.